Hello dear students, good afternoon all of you. In last lecture we have studied about the enzymes. In last lecture we have studied definition of enzymes and how what is the nature of enzyme. So I told you nature of enzymes is proteinaceous. So most of the enzymes are proteins, but not all proteins are the enzymes that we have discussed in earlier lecture. So little bit information was remaining, and that I am going to tell you that. See, in 1833, in this year, this scientist Anselm Payen, they discovered first enzyme. So first enzyme to be discovered by this scientist is diastase. At that time he given name enzyme, that is diastase. Today this enzyme is known as amylase, which is important in digestion of starch, breakdown of starchy food material. So in this year the Anselm Payen discovered the first enzyme, that is diastase. At that time he not use the word enzyme, just given directly the name diastase, but they were not aware about that is actually enzyme. Then in this year 1877, this enzyme, the term used first by Wilhelm Kuhn. This Wilhelm Kuhn was studying about fermentation reaction. So you might be knowing that fermentation, these are the reactions by which alcohol, many organic acids are prepared. You these days you people are knowing that is importance of sanitizer, that is approximately having 70% alcohol. So that alcohol, ethyl alcohol, especially ethanol, that is IUPAC name, ethanol, that is being prepared by fermentation of sugars, molasses by yeast, Saccharomyces cerevisiae. So these scientists were studying on these reactions going on inside of the cell, in vitro, in vivo. So they studied the enzymes, different type of reactions, biochemical reactions inside the yeast, and this Kuhn first used the term enzyme. This is from Greek, en means inside and zyme means yeast. So these were firstly discovered in the yeast, that is unicellular fungus. So these are the things which you should know in an introductory part of the topic of enzymes. So let us study another point from our syllabus. So this was all about the introduction. Now we are moving towards a little bit topic which is out of syllabus actually, but that is important for our knowledge. So naming an enzyme, how names are given to the enzymes, that is also an important. We will finish this in little bit minutes in few minutes. So common names to the enzymes give little information about enzymes. There are many enzymes which are given common name, like that of trypsin, chymotrypsin. These are enzymes, but these are not showing you that where it is synthesized, or what is actually function of this trypsin. That is because this is common name. So common name does not tell you more information or thorough information of that. For example, trypsin. Trypsin is a proteolytic enzyme which degrades proteins or break protein molecules. So trypsin is a proteolytic enzyme which is secreted by pancreas. But by the name trypsin we are not getting any information. That you have to play with that. You have to remember that trypsin is a proteolytic enzyme. So you are getting no logic about what is trypsin. It is enzyme, but which type of enzyme, what is its function. So you have to remember that it is proteolytic enzyme which breaks protein molecule and that is secreted by pancreas. That we have to remember. That is the drawback of common names. एंजाइम कमीशन के बारे में बात करेंगे आगे जाके. So many enzymes are named according to their substrates. Nowadays in a science, in a metabolism, biochemistry, so many enzymes are named according to their substrates. So on which substrates enzymes are acting and in which reaction enzymes are acting. So those enzymes are named according to that reaction and according to that substrate. So many enzymes are named according to their substrates and reactions which they catalyze. In this system, new system, suffix ase, any enzyme you will get scientific name or enzyme commission name. ASE is there, any enzyme like that of diastase, amylase, phosphorylase. You might have heard about that in your previous classes. There are varieties of enzymes in biology. So suffix ase is added according to the substrate and reaction. Whatever the substrate is there and what is the reaction in which enzyme is involved, that enzyme is named according to that substrate and reaction. Simple case is there, no rocket science. For example, ATPase, ATP synthase, phosphorylase, these are some examples how ASE suffix is coming. See, here is ASE, ASE, ASE. So in modern concepts, ASE is a suffix added according to substrate and reaction. Enzyme commission, that is standing in a biochemistry, in metabolism, metabolical science. So enzyme commission gives standard names. Enzyme commission like that of in chemistry, that IUPAC name is there. Like that of in this biochemistry, for enzyme there is enzyme commission and standard names are given to the enzymes. For example, hexokinase. Hexokinase is an enzyme and this is given EC 2.7.1.2. These numbers, what does it indicate? This 2, 7, 1. These are major categories, major classes. So this first one 2 is a class, second that is 7 is a subclass indicating subclass, then one number is coming, 1 is subclass, and another last number is 2, that is the serial number. Serial number of enzyme under the subclass, under which subclass that is added. So you should be clear with this. This is a basic knowledge. This is not a part of syllabus, but we must know actually how enzymes are named. For systematic classification, now we are moving towards the classification of enzymes, and that is a systematic one. For systematic classification of enzymes in biochemistry, enzyme commission has classified these enzymes into 6 classes. Don't forget this, there are 6 classes. So let us go with the topic classification of enzymes. I was telling you that there are 6 classes of enzymes. So we are going one by one. So first class, please be concentrated on the topic. First class is oxidoreductases, EC1 enzyme class 1, that is oxidoreductases, oxidoreductase. So from this word you might be getting something, some information, that is oxidoreductase. Oxido means oxidation, reductase means reduction. So you are getting some information from this. So that is a standard value, standard nomenclature, standard classification. They catalyze these enzymes coming under this class. They catalyze oxidation reduction reaction. Simple, no rocket science is there. By the name we are remembering all the things. Just you have to remember names of the enzymes, examples of the enzymes. So first class is of oxidoreductases. So they are catalyzing the oxidation reduction reactions. So we can represent reactions like this. So A is one substrate, B is another substrate, but A is in oxidized state, B is in reduced state. So we are using enzymes and reaction is taking forwardly. So what is happening? A is getting reduced form and B is getting oxidized form. So this change has been taken place. This is reaction of oxidation and reduction, simultaneous reactions are there. So A पहले तो ऑक्सीडाइज्ड स्टेट में है, वो बाद में चला गया रिड्यूस्ड स्टेट में जब रिएक्शन पूरा होता है. सो दैट इज रिडक्शन. Simple is that. So you might have studied this in your chemistry classes. There is no rocket science. These are simple reactions, but you have to remember throughout the topic. For example, let us consider examples. Oxidases. So oxidases, these enzymes use oxygen as an electron acceptor, but they are not adding that electron into the substrate. You must remember this. So oxidases, these are using oxygen as an electron acceptor. Dehydrogenases, so they use molecules other than oxygen as an electron acceptor, like that of NAD plus, nicotinamide adenine dinucleotide. So they will be using NAD plus, NAD as coenzyme. Oxygenases, they directly incorporate oxygen into the substrate. So these are oxygenases. These are examples coming under this class. Peroxidases, you might have studied in practical classes, enzyme peroxide study. So peroxidases are also there. They use hydrogen peroxide H2O2 as an electron acceptor. So H2O2 is getting reduced, whatever things are going on there. So this is all about first class of enzymes, that is oxidoreductase. Then next class is second class, transferases. So next class is EC2, enzyme class 2, transferases. So you might be getting some information from. So they are transferring groups, molecular groups, functional groups. So transferases. So they catalyze reactions that involve transfer of groups from one molecule to the another. एक मॉलिक्यूल से ग्रुप को लेंगे, एक केमिकल ग्रुप होगा तो वो दूसरे के ऊपर ऐड कर देंगे, इतना सा रिएक्शन है. ट्रांसफर ऑफ ग्रुप्स. Examples of such groups include, which type of groups are transferred? Carbonyl group, carboxyl group, amino group, methyl group, phosphoryl group, acyl groups. So these are groups which are being transferred by these enzymes. So we can general reaction we can represent like this: A B. There is bonding between A and B, and they are getting reacted with C. So enzyme is working here, and what we are getting actually, so A is getting separated, A is getting separated, and bonding between B and C is made there. So there is transfer of group from this B to C. B group is transferred to C. Examples, so many examples are there. I have taken a few of them. Trans carboxylases, you have to remember at least 2-3 examples from this. Trans carboxylases, trans, that means transfer. Trans carboxylase, that is transfer of carboxyl group. Here is somewhat spelling mistake is there, please. Carboxyl group, carboxylate group actually. So R is coming there, E instead of carboxylate. I have written spelling here. So transfer of carboxylate group to substrate. सबस्ट्रेट में ये डालेगा कार्बोक्सिल ग्रुप. दूसरा है ट्रांस एमिनेजेस, दैट इज ट्रांसफर ऑफ एमिनो ग्रुप, NH2 group, you are knowing that amino group. Kinases, that is transfer of phosphate. Kinases are transferring phosphate group from ATP to the substrate. These are substrate, you are knowing. Substrate means reactants. So kinases are adding phosphate into the substrate from the ATP. Phosphorylases, so transfer of inorganic phosphate from substrate to substrate. So this was all about second class of enzymes, transferases. Next class coming is hydrolases. Hydrolase, so you have to remember this thing, you have to write down these things whatever going on in lecture. You can repeat it for 2-3 times and you will get clear with that. So let us study what is hydrolases. They catalyze the reactions in which the cleavage of bond is accomplished by addition of water. So bond cleavage means breaking of bond is there, but that is accomplished by addition of water. Water is important, H2O. So water is important there, and name of enzyme is hydrolase. Class of enzyme is hydrolase. So reaction we can represent is like this: A and B. Here is bonding between A and B. These are reactants. So what is happening here? H2O is added. Here enzyme is working. Whatever enzyme, general reaction is there. So there is bonding taking place between A and H, that is H2O. We can split it into HOH. So bonding between A and H and B and OH is made here. So this is simply cleavage. So cleavage of bonds by using water molecule. Examples, let us see examples. Phosphodiesterase. So phosphodiester bond is there in nucleotides like the nucleic acids, DNA, RNA. There is phosphodiester bond between two DNA molecules between adjacent nucleotides. So that bond is broken by these enzymes phosphodiesterases. So they cleave phosphodiester bond. Phosphatases, so they remove phosphate from the substrate by using H2O, water. Peptidases, so they cleave peptide bond, that is amide bond, which is present in proteins, amino acids. Two amino acids are joined by this peptide bond, amide bond. So this was all about hydrolases. So let us move to another class of enzymes, that is lyases. You might be getting boring with this classification, but this classification is a standard classification that is important in biological sciences. You have to get it repeated. प्लीज़ रिपीट दिस, रिपीट करना पड़ेगा आप लोगों को, रिपीट करके ये ध्यान में रह सकता है। ऐसे ही एक बार पढ़ लिया और ध्यान में रह सकता है तो हम स्वामी विवेकानंद नहीं है। प्लीज़ बी कॉन्सेंट्रेटेड ऑन द टॉपिक। सो enzyme class 4, that is lyases. They catalyze breaking of carbon carbon, carbon nitrogen, carbon oxygen, carbon sulfur type of bonds. So they are having ability to catalyze the reactions in which breaking of these bonds is taking place. Lyases is the class of enzyme which is involving enzymes which break these type of bonds. You might be knowing these bonds are there. कार्बन कार्बन के बीच में बॉन्ड होता है, कार्बन नाइट्रोजन, कार्बन ऑक्सीजन, कार्बन सल्फर, तो कोवेलेंट तरीके के बॉन्ड्स होंगे, कोऑर्डिनेट तरीके के बॉन्ड्स होंगे, व्हाटएवर the type of bonds you have studied in chemistry. So these bonds are broken by means of other than hydrolysis or oxidation. So here is not taking place hydrolysis. So we have studied that in hydrolases. Not taking place oxidation, no oxidation, no hydrolysis, but bond is broken. So that is enzyme involved coming under class lyases. The bonds are cleaved by process of elimination. So elimination reaction होती है इसमें, please remember. So the bonds are cleaved, broken by process of elimination reaction. Elimination reaction is there. So generally we can represent this A, B, here is double bond between A and B, these are reactants, and H and X. So this is another reactant and enzyme is working under this. So what is being eliminated? Let us see, A is getting bonded with X. So A get bind with X and B get bind with H. So products be eliminated here. Examples, let us see examples. Aldolates. So this is a famous example, aldolates coming under lyases. Removal of aldehyde group or aldehydes via elimination reaction. Elimination type of reaction that you have studied in organic chemistry paper in second year of you will study this actually in your coming chemistry lectures. So aldolases, you also have studied this in your 12th standard, aldolates, in your photosynthesis respiration reactions. So the removal of aldehydes via elimination reaction. Next example is synthases, not synthetases. Remember this. This is synthases. So they link two molecules without involvement of ATP. No ATP is involved in this reaction. So they join molecules, two molecules without involvement of ATP. Next next example is dehydratases. So dehydration reaction. Dehydratase is the enzyme. So that is removal of water H2O via elimination reaction. Elimination reaction होगा, water बाहर निकलेगा. Decarboxylases, that is removal of CO2 via elimination reaction. This was all about enzyme class 4, that is lyases. Lyases. Please remember. Now we are going to the enzyme class 5, isomerases. Isomerases. They catalyze several types of intramolecular rearrangements. You are knowing isomers. Isomers. That is, chemical formula is same or molecular formula is same, but structure is different. That are isomers. So they catalyze these enzymes coming under isomerases catalyze several types of इंट्रामोलिकुलर रीअरेंजमेंट्स मॉलिक्यूल का जो इंट्रा अंदर का अरेंजमेंट है उसमें चेंज लाएगा, कैटालिसिस होगा इसका एंड यील्ड आइसोमेरिक फॉर्म्स. तो आइसोमर्स बनते हैं. दे these reactions or these enzymes take part in the reactions which prepare isomers of molecules. So these are reactants A and B. So bonding is A to B. Now here bonding is B to A. So direction of bonding or rotation of bond is specific. So that I have changed, but overall molecule is remaining same. Molecular structure is changing. So you have to remember this. Rearrangement. Atomic arrangement, molecular arrangement is changing inside the molecule. Examples देखिए क्या क्या है. Mutases. Mutase is a group of enzymes. So they catalyze intramolecular transfer of functional groups. So functional group का transfer होगा एक ही मॉलिक्यूल सेम मॉलिक्यूल के अंदर. ये इंपॉर्टेंट है, ये दूसरे मॉलिक्यूल के ऊपर लेके नहीं जा रहा है, सेम मॉलिक्यूल के ऊपर ही अरेंजमेंट होगा. सिस ट्रांस आइसोमरेज दैट you might have studied in your 11th 12th standard, cis trans isomerism. So cis trans isomerase is the enzyme example coming under this class. They catalyze the isomerization of geometric isomer. Geometric isomers, you might be knowing geometric isomers. So they are going to prepare geometric isomers. Epimers catalyzes inversion of asymmetric carbon. जो कुछ भी asymmetric carbon होगा उसका inversion उल्टा किया जाएगा उसको, तो इसको एपिमरेज कहते हैं एंजाइम को. रेसिमेजेस दैट इज मेकिंग रेसिमिक मिक्सचर D and L type. So they interconvert L and D isomers. L का कन्वर्शन D में होगा, D का कन्वर्शन L में होगा, कुछ भी हो सकता है. This was all about enzyme class 5 isomerases. Next, last class coming is ligases. You might have studied this in your DNA replication in 12th standard DNA replication or in 11th standard maybe. Ligase, that is joining DNA molecules by using ATP. Let us see. So they catalyze formation, not breaking, please remember, formation of carbon-carbon bond, carbon-oxygen bond, carbon-sulfur bond, carbon-nitrogen bond. So this type of bonds they are making. So they catalyze formation of this type of bonds with simultaneous hydrolysis of ATP. एक बाजू को ATP ब्रेक होगा और दूसरी बाजू को ये बॉन्ड फॉर्मेशन हो जाएगा. Simple reactions are there. So generally how we can represent this as A is one reactant, B is another reactant, and simultaneously we are using ATP. So A and B के बीच में बॉन्ड बन रहा है, ATP ब्रेक हुआ, इसके लिए उसका ADP तैयार हुआ और इनऑर्गेनिक फॉस्फेट भी रिलीज होगा. ये आप दिखा सकते हो, नहीं दिखाओ तो भी चल जाता है. चलो ठीक है. एग्जाम्पल्स देखो इसका. Synthetases, not synthesis. So synthetases are coming under enzyme class 6 ligases. synthesis of new molecules. Whatever molecules are there, newly synthesized molecules are there. Next example is carboxylases. So that is adding CO2 as substrate. So that enzyme carboxylases is adding CO2 as a substrate. So this was all about your enzyme classification. This is a standard classification from the Enzyme Commission. Like I was telling you, in chemistry IUPAC nomenclature or IUPAC is there, International Union of Pure and Applied Chemistry. Like that of here is EC, Enzyme Commission. So this Enzyme Commission has given this classification. This is standard one. If you Google it, you will get more information from this classification. So simply how you can remember this word by OTHLIL, oxidoreductases, transferases, hydrolases, liases, isomerases, ligases. So go through this OTHLIL and remember. This is easy to remember. So let us stop here, and remaining part we will discuss in upcoming lectures. Thank you one and all. Thank you very much.